Two heads are always better than one, they say, which is especially true when it comes to solving crimes. We can see it in one of the most iconic crime-fighting duos out there, Sherlock Holmes and Dr. Watson. Each of them bringing their own strengths to the table, but together, they're able to solve the most complicated mysteries in London, catching clues that the other one might have missed. What if I told you this was also true for cancer drugs? By having two therapeutic agents, you're able to catch the cancer cells that one of them might have missed, and thus combating resistance. Now imagine having the best parts of Sherlock Holmes and the best parts of Dr. Watson together in one person, one super detective. My research looks at the use of a single agent, two-headed cancer drug called the KT3000 series for a treatment of a criminal that targets children. This criminal lives in their bones and often finds hiding places in other organs. And once this criminal has established its hiding places, the chances of survival are very low. Now, who is this mystery criminal, you might ask? Well, it's Ewing sarcoma, my dear Watson. Now, how does this drug work? Well, each of the heads of the drug hones in on a particular protein of interest, stopping them in their tracks. And the way that it does this is that it binds to and blocks these little pockets of activity within the protein. One targeting PARP, the other HDAC, vital in DNA repair and gene expression, respectively. Cancer cells use these proteins in order to bypass cellular checkpoints in order to grow and build empires undetected. By targeting PARP and HDAC with these inhibitors, you're able to trigger biological clues like induction of cell cycle arrest or activation of DNA damage markers that signals to the other pathways around them that something here isn't quite right, and later sentencing the cell to death so that it can never strike again. We can attach fluorescent markers to these biological clues in order to visualize and measure them. The more biological clues that we can detect, the better the drug. Using our own methods of deduction, we're able to show that in both two-dimensional and three-dimensional cancer cell models, that at the same concentrations, our two-headed drug can outperform current clinically approved PARP and HDAC inhibitors, both as single agents and in combination. We are currently investigating the mechanics of our drug in hopes to improve it, so we can hopefully send it to clinical trials later. And that way, we can give Ewing sarcoma its last bell. Thank you.